a little quickly through this. You will want to write a lot of this down. We're going to hit a lot of vocabulary words again. So if you're writing on paper or whatever, that's fine. But when we have these two, and they're going to reproduce, we're going to look at one trait at a time. There. I'll make their teeth look a little bit better. So we've got Bucky, we've got Becky, and let's look at their eyes. So they're both brown-eyed beavers, which is their phenotype. Remember that? So we're going to start off, we're going to say that Bucky and Becky are both what for eye color? Do you remember this term? They have different alleles. If you're interested in kissing somebody whose genitals are different from yours, you are what sexual? Heterosexual. These guys have two different alleles. They are heterozygous for this trait. Now they're heterozygous, which means they have a copy of the dominant allele and a copy of the recessive allele. The dominant characteristic here is brown eyes. So both of these beavers are going to have brown eyes, yet they're both carrying a hidden recessive for red eyes. So let's think about Bucky and Becky making sperm and eggs. Yay! So we'll start off with Bucky. Every time Bucky's body makes sperm, remember we went through that process, we started off with a diploid cell, we went through a bunch of different divisions, we ended up with a cell that had half the normal number of chromosomes. That cell's a what kind of cell? Haploid. It's a haploid cell. Happy little haploid cells. So every time Bucky makes a sperm, there's a 50% chance that it's going to get what? Look at the two alleles he has for this trait. 50% chance that each sperm would have a big B. 50% chance that it would have a what? Little b. So roughly half his sperm we would expect to have the dominant allele, roughly half his sperm we would expect to have the recessive allele. Now we get to Becky and her eggs. There's a 50% chance that any egg that Becky makes will have which one? Big B. 50% chance any egg she makes will have the little b. And this is all chances. You know, in general, we're going to expect that about 50% of her eggs will have the dominant, 50% will have the recessive. We can figure out the percentage chances of their offspring having a particular combination of genes and having a particular appearance. And that's all it is calculating odds. So a Punnett Square is the tool we use to figure out those percentages. And we're going to do one just sort of free form, then I've got a packet for you where you're going to do some monohybrid Punnett Squares. So most commonly we put mom's contribution across the top and dad's contribution down the side. So the maternal contribution, Becky's eggs, we figure are a mix of big B and little b. Bucky's sperm, we figure are a mix of big B and little b. How many of you remember clearly having done one of these before? Okay. A few of you are raising your hands who weren't earlier, so maybe you forgot what it was called. This is a monohybrid Punnett Square. Mono means how many? One. We're crossing one trait at a time. We're working with one trait at a time. So to finish this, you just pull these down. Big B, big B, big B, little b, big B, little b, little b, little b. That represents the odds of any one of their offspring having a given genotype, and we're going to talk about phenotypes next. So remember, genotype is the type of genes you have. And when we do this, we're really interested in the ratio of one genotype to another in their offspring. So the first thing we always do is a genotype ratio. Okay? possible genotypes that exist for this trait. We could have big B, we could have big B, and we could have little b, little b. If you've got something that has two alleles, a simple dominant recessive, you're always going to have three possibilities for genotype for that gene. So we're just going to look down here. How many big B's do we have? Big B. There's one. Do we have any others? Nope. Just one. How many big B, little b's do we have? 1, 2. And little b, 1. So we say that for this trait, we would expect to see a 1 to 2 to 1 ratio in the offspring. And any time we cross two heterozygous individuals, that's what we get, is that 1 to 2 to 1 ratio for a single trait. Now, so genotype is the type of genes you have. Phenotype is the physical appearance of the organism. So for this trait, eye color, let's do our phenotype ratio. There are two possibilities for phenotype. Give me one of them. Red eyes is the recessive. We usually list the dominant first, just a matter of course. Pardon? There you go. So our two possible phenotypes are brown eyes or red eyes. And what we can do is look at each genotype and say, well, what color eyes would this one have? So big B gives you what color eyes? Brown. So that's 1 for brown. Big B, little b gives you what color eyes? Brown. That's 3 for brown. And little b gives you, it's recessive. You got 2 copies of the recessive, you get the recessive characteristic. You get red eyes. So what do we see for a ratio? We see a 3 to 1 ratio. Well, what that really means, so does that mean that if this beaver couple had a litter of 4 kits, we would see 3 brown-eyed individuals and 1 red-eyed? No. This is just the odds. For every conception that occurs with their eggs and their sperm, so we would expect that about 75% of the offspring that were conceived, 75% of the fertilized eggs would end up having a genotype which would lead to brown eyes. About 25% of any offspring from this couple we would expect to have red eyes. Now, could they have 4 red-eyed babies in a row? Yeah. Of course they could. Let's do a quick one for sex. So mom contributes an X and an X. Dad contributes an X and a Y. XX, XX, XY, XY. Gesundheit. 50% male, 50% female, on average. Who here has at least 1 sibling? Who here has exactly 1 sibling? Okay, so since you guys are girls, all of you have a boy sibling, right? Because it's 50-50. So if you have 1 boy, you automatically have to have 1 girl, right? No. The odds at each conception go back to 0. So the odds of the previous conception don't affect the current conception. Every single one is a brand new roll of the dice. You can get anything. It's amazing. So yeah, and the examples that we're doing, we tend to simplify stuff in high school biology. I mean, we're starting, in the middle school, it's really grossly simplified. Here's where we start. Who was it who said the one time? Every year they tell you a little bit more truth. You know, in elementary, it's all sunshine and roses, and by middle school, we get a little bit of the darker side of things, and by high school, we're telling you more truth. We've grossly simplified genetics for you up to this point. I'm telling you, it is more complicated. There are a few traits that are monogenic, and those are the ones that are easiest to work with. So, okay, so I'm going to hand out a packet for you. Get your 3 characteristics recorded. Get the mother's genotype and phenotype, father's phenotype for 3 traits. Okay. Are traits passed on one at a time? Is there a sperm and an egg that has one allele for each trait and they somehow all magically fuse? No. A single sperm contains 1 copy of all the possible genes. A single egg contains 1 copy of all the possible genes for that species. So if we're going to go back to Bucky. Okay, so what we just did was a monohybrid cross. That means we crossed a single trait at a time. Well, now we're going to do a dihybrid cross. Flip the page. Di meaning how many? 2. We're going to cross 2 traits at a time. In reality, every time a species reproduces, you're crossing like 30,000 traits at a time. It's a lot of combinations. That's in humans. I don't know how many genes beavers actually have. We're showing 10 genes here. So let's say we've looked at tail length and eye color, and Bucky and Becky are both heterozygous for both traits. So every time Bucky manufactures sperm, there's a 50% chance his sperm will have a big B, right? 50% chance, we already established that. 50% chance his sperm will have a little b. Well, there's this thing called independent assortment. It means that traits usually aren't linked together. A lot of traits are not linked together. So we're going to assume that these are independently assorted. So for every big B sperm that Bucky produces, there's a 50% chance it'll get a big T. But there's also a 50% chance it'll get a little t. And for every little b sperm, that Bucky produces, there's a 50% chance it'll get a big T, and a 50% chance it'll get a little t. We sometimes do this as a probability chart, and that's what's shown on your paper, where we say, okay, well, 50% chance of a big B or a little b. For every big B, there's a chance of a big T or a little t. For every little b, there's a chance of a big or a little. This is one possible combination. This is another. This is another possible combination. That's another. We have 4 possible combinations for his gametes. Does that make sense? Okay. Well, now, I screwed that up. Now, for Becky's eggs, what are the odds? What are her eggs going to look like? They have the same genotype. Guess what? The odds for her eggs are identical. So in the monohybrid cross, we crossed 1 trait at a time. Now we're going to cross 2 traits at a time, and it's going to require a larger chart. So I'm going to shrink the eggs and sperm here. And you should be copying this down on the sample page when we get to it. So we have, 1, 2, 3, 4. 1, 2, 3, 4. And there is a sample page right on the packet there that I want you to make sure you do this in. So we're going to put maternal contribution across the top. I have big B, big T, big B, little t, big T, little b, little t. And those are just the egg combinations that we know exist for Becky Beaver. Down the side, looking at the paternal contribution, we have big B, big T, big B, little t, little b, big T, little b, little t. going to do the same pulling down thing that we've done with all the rest of these. The thing you have to be careful about with this one is you have to put the alleles for each trait together, because we're looking at the allele combination that they got for each trait. This is like watching paint dry, I know. It's not all that exciting. What is exciting is what you can do with the information. Because it gives us the power to predict a lot. Whew! Okay, that's a whole lot of squares. To Punnett Square, we always want to look at some ratios. And with a dihybrid Punnett Square, we actually have 9 different genotypic combinations that we could see. So, first of all, we've got big B, big B, big T, big T. Well, then we have big B, big B, big T, little t. And, oh yeah, we have big B, little t, little t. Now, if you're working on Doceri, I'm going to copy those 3. You're going to see why in a second. So I could also have big B, little b, big T, big B, little b, big T, little t, big B, little b, little t. It sounds like a Dr. Seuss book. And then I could get little b, big T, little b, big T, little t, little b, little t, little t. Now, when you tally these up, it takes a... do you? Okay, when we tally these, it really helps to mark them off. I see 1 of these. I see 2 of these. I see 1 of these. Hey, 1 to 2 to 1. That's weird. I see big B, little b, big T. I see 2 of those. Big B, little t, heterozygous on both traits. 1, 2, 3, 4. Heterozygous for eye color, homozygous recessive for tail length. I see, whoops, let me do that in another color. 1, 2. Little, big, I see 1. Little, big, I see 2. And homozygous recessive for both traits, I see 1. That's horrible. Absolutely, utterly awful. But that's okay. Now, could we translate that? How many phenotype combinations does this boil down to? We could have brown eyes with a long tail, dominant, dominant. We could have brown eyes with a short tail, dominant, recessive. We could have red eyes with a long tail. We could have red eyes with a short tail. Whoops, there are only 4 phenotype combinations for this. I'm going to bring these and shrink them just a little bit. So there are only 4 phenotype combinations for all those genotypes. And when you're tallying these up, again, it's really, what I actually very often do is I go through with a highlighter. So let me see, everybody who's got brown eyes, okay, all of these have brown eyes. Now, which one of those have a long tail? These have long tails, and these have long tails. So 2, 4, 8, 9. So I have 9 individuals with brown eyes and long tails. These guys have brown eyes but short tails. That's 1. These guys have brown eyes but short tails. So I have a total of 3 who got the brown eyes but got short tails. Now I know that everybody down here, these are my red-eyed babies. How many of those red-eyed babies have long tails? These guys have long tails. These guys have long tails, so I have 3. And I have 1 poor little baby. This is that one kid in the family who gets picked on, the one beaver baby whose siblings make fun of it because it not only has red eyes, but it got the short stumpy tail. Poor beaver baby. Now, again, these are percentage odds. These are ratios. Out of 16 possible genotypes and 12 possible, no, 9, I'll get it yet, good Lord, it's that kind of day. Out of 9 possible genotypic combinations and 4 possible phenotypic combinations, that's just a percentage odds on any one of them happening. What you have for homework is this. Finish the 3 monohybrid squares. Okay? So I want you to do 3 monohybrid squares. I want you to attempt 1 dihybrid on your own, using traits from your own beaver babies. So you've got the mother and father's genotypes. So I want you to try it, and then we'll look through it tomorrow.